I know that these textbook looking diagrams can seem a bit vague, like what's an inhibitor, where does it come from? So we know that normal enzyme substrate reactions, the enzyme combines with the substrate binds into the active site, the enzyme modifies the substrate in some way, and then spits out the products, right? And the enzyme gets regenerated. So the enzyme doesn't actually undergo any permanent change in its process. But if we then introduce something else into the system, which looks like the substrate, but isn't the substrate, which we call an inhibitor, in particular, this is called a competitive inhibitor. This inhibitor could lodge itself into the active site and exclude the substrate from binding into the active site. So this is irreversible binding into the active site, prevents the substrate from binding and prevents the enzyme from modifying the substrate into the products, and we get no reaction. We then have non-competitive inhibitors where the inhibitor binds to an alternative site on the enzyme. What's this site called, do we think? Do you remember the name for that site? So this one here is the active site because that's what binds to the substrate. But then we got another site on the enzyme. Do you remember what that's called? This here is called an allosteric site. It's a site of regulation of the enzyme where we can bind a non-competitive inhibitor. It's called, why would it be called a non-competitive inhibitor?
It doesn't take over the active site.
Yeah, it doesn't compete for the active site. It binds to an alternative site, which can still render the enzyme non-functional. So the inhibitor binds the alternative site, and that in turn changes the shape of the active site because we've got something lodged into the site here. And now the substrate is no longer complementary to the active site, can't bind, and we don't get a reaction. So this phenomenon in which we bind a small molecule into one domain of the protein to change the shape at another domain, that phenomenon is called allostery, and that's why this is called the allosteric site. Okay, this is some new slides. So let's think about some examples. I chose the example of, so I guess this principle with inhibitors is that drugs often act as enzyme inhibitors. I think it's something like 80% of drugs all act as inhibitors of key enzymes in the body. So one class of medication is called statin medications. These medications are used to lower cholesterol in the body. So these medications function to basically inhibit one of the key reactions in the pathway to synthesize cholesterol in the body. So this is the reaction that happens. We convert this species here to that one there. And that reaction is catalyzed by the enzyme called HMG coenzyme A reductase, right? That's the enzyme. And statin medications are potent competitive inhibitors of HMG coenzyme A reductase. If I inhibit this enzyme in this pathway, then the pathway can't continue. So the statin medications act as a competitive inhibitor of this enzyme. If I competitively inhibit this enzyme, I prevent this reaction from happening, and hence I prevent all the subsequent reactions from happening because I don't have any of this. Does that make sense? Cool. So yeah, some notes there. Nice. So this is what the enzyme looks like, HMG coenzyme A reductase. Likewise, we can try and find some secondary structures in here. So here we have some alpha helixes. We've got multiple alpha helixes because this protein is actually a transmembrane protein. So we've got these many alpha helical transmembrane segments that go up and down, up and down, up and down in the membrane. But we also have some beta sheets. They're sort of hiding in there. There's one there, which is sort of hiding. So since the reaction catalyzed by HMG coenzyme A reductase is the rate limiting step in cholesterol synthesis, this enzyme represents the sole major drug target for contemporary cholesterol lowering drugs in humans. So this step here is the rate determining step of this pathway. What that means is that the speed in which this reaction happens determines how quickly the entire pathway happens. So that's why this enzyme here is of particular interest to inhibit that enzyme. Just some more information. It's got 888 amino acids. Nice. So what do these statin medications look like? There's many different types of statin medications where they all act as competitive inhibitors of the HMG coenzyme A reductase enzyme. So these are all administered to individuals with high cholesterol to inhibit the natural synthesis of cholesterol in the body. So individuals with high cholesterol take these. Let's think about one example. So one example is atorvastatin. I don't really know how to say it. So So like all statin medications, this medication works by inhibiting the HMG coenzyme A reductase, which is an enzyme that's found in the liver that plays a role in producing cholesterol. It's a competitive inhibitor of the enzyme. However, unlike all of the other statin medications, this one's actually a synthetic drug. So they've just synthesized it in the lab, opposed to being derived from some natural product or something like that. Cool. So that's what it looks like. Here's a more friendly biology looking diagram just to show its shape, I guess. So how does it actually inhibit the enzyme? So we say that it's a competitive inhibitor. I know that a lot of the people that attend my sessions do chemistry, so I will talk about some chemical properties about how it works. So we think about the enzyme. I'm pretty sure the active site is somewhere in the middle here. So this is taking a close-up view of the active site, and it's showing just the amino acids that are sort of in the cavity of the active site, and it's showing how those amino acids bind to the bound statin drug in the active site. So we see, this is sort of simplifying what this diagram here is showing. So we can see this is our drug. We would have some hydrophobic interactions between this small alkyl chain here. So this hydrophobic group here would interact with some hydrophobic amino acids in the active site. This charge group up here would probably participate in some sort of hydrogen bond or ion dipole bonds in the active site. This electronegative fluorine atom would be interacting through a hydrogen bond with some of our amine residues in the active site as well. And likewise, this oxygen of the amide linkage is interacting hydrophilically with a hydrophilic serine amino acid in the active site as well. So you can see how the spatial arrangement of the amino acids in the active site, as well as the chemical nature of the amino acids in the active site, all facilitate the sort of perfect binding of the inhibitor into the active site. So they must have designed this drug to facilitate the binding of different components into the small, to fit and bind into the active site sort of perfectly, right? So you want to facilitate all these different interactions by designing the drug perfectly. So that would, of course, require an understanding of the three-dimensional structure of the amino acids in the active site to then design a drug that would bind into the active site and have those particular interactions. So that's like a bit of a more complex look at what inhibitors actually are and what they do compared to looking at something like this. So I thought that these were a bit inadequate.